All right, so I'm going to talk about our work using C. elegans. There we go. Okay. So, honestly, this kind of a talk is a little bit of a challenge because I know we have parents in the audience who aren't scientists. We also have scientists here. I've tried to gear this so that everyone can understand it, and I'm happy to answer questions that are the most basic kind of questions as well as more technical questions at the end. So, what I want to talk to you about is like our model. So, we use worms to study ADSL deficiency. Why do we use worms and what are our strategies? I'm going to talk about three different phenotypes that we have discovered in our ADSL worms and then what we've learned about that. And in the end, I am going to discuss our work where we are taking the potential therapeutics that were found in the Perlara screen, which I'm assuming most people know about, but I'll talk about that and show where we are with what we have shown that those can do in our model. Okay, so we use this microscopic nematode, and this movie here is showing you what the a little worm looks like. They're about a millimeter long. The reason we use them is that we can do genetics, we can do experiments, we can do a whole lot of stuff with these animals. And they also have purine biosynthesis that's just like what you see in humans, right? So this pathway you've seen before, and C. elegans has the exact same pathway, conserved ADSL gene. You might think C. elegans are really simple, and actually we use them because they are, but they also have a nervous system, a musculature, right? They have intriguing behaviors, and so we can study all of this kinds of effects of ADSL in the nervous system and in the musculature, for example, in these worms. So what we do is we take the normal worm. Again, I'm showing you a normal worm just swimming here in some buffer. We remove ADSL function, and so we have animals that do not have any ADSL function. That right there is a little bit unusual compared to in humans, for example. We know that how much ADSL activity is left will determine the severity of the disorder. And here we have worms that are alive. There's a lot of things wrong with them, but they're alive without ADSL. And so, anyway, we use that most severe form to try to figure out what's going on. We want to know what's wrong, why does it go wrong, and what can we do about it? Okay, so um, we've already found a lot of different phenotypes, and I've noted some publications down here that describe what's wrong with ADSL worms. And on the right here, you're looking at one of these ADSL worms compared to on the left, a normal worm. So what you might notice, first of all, is they're small. All right. And they're also slow in the way that they swim. We've shown that they're uncoordinated. They have learning deficiencies. They have defective signaling at their neuromuscular junction. They're also infertile and they develop very slowly. It takes them more than twice as long to get from being an embryo or an egg that just hatched essentially to an adult. Okay, you've also seen this slide before today, right? So I said that metabolism is the same in C. elegans, purine metabolism as it is in humans. So ADSL acts twice in this pathway. And we are doing our experiments under a couple of different overarching hypotheses, again, that you've already heard about today. One is that when ADSL is gone, you get this buildup of SAICAR that's hypothesized to be toxic, right? And the other is when ADSL is gone, you really are interfering with purine biosynthesis. And we believe that interference with purine biosynthesis and homeostasis also is a problem, and that each of these can cause different phenotypes. And actually, again, in our published work, we have shown this. We have shown that there's toxicity of SAICAR and that some of our neural and behavioral phenotypes are because of that. And we have also shown that purine biosynthesis, the homeostasis of purine biosynthesis underlies, for example, some of the defects that we see in oogenesis in the animals. So both things are at play here. And part of what we're trying to do is map all the phenotypes to these different areas so we understand the effects of ADSL, for example, in muscle or in neurons, like I have been using as examples. Okay, so I'm going to tell you about a couple of these neural and behavioral kind of phenotypes that we know are due to SAICAR toxicity and what we've learned there. So first, just to explain one of these neural phenotypes, our animals don't learn properly, all right? And this slide here is used to explain what I mean. What you're looking at is a culture plate, right? That's divided into 4 quadrants, and we've placed a certain concentration of salt in two of these quadrants. We put animals in the middle of the plate. They have access to all four quadrants. We give them a half an hour and we see what they do. So it turns out that C. elegans animals like salt. And so we give them a half an hour, they're going to all go to where the salt is because they like that and most likely associate it with food. However, if we take normal animals and we put them in salt, all right, but we also pair a negative cue with that, we starve them, all right? So we starve them for 15 minutes in salt. And then we repeat this assay where we put them on the plate with the four quadrants. They learn not to care, right? They don't prefer salt anymore because they've been starved in the presence of salt. And so they learned, well, salt wasn't that great after all. So they just kind of ignore it. ADSL animals are perfectly capable of sensing salt and responding to it, right? They act just like normal animals when you do this assay at first, but after we starve them for 15 minutes in the presence of salt, their behavior is different than normal animals. They actually actively avoid the salt. So they learn to hate the salt, essentially, all right? And so this response is, it's not a lack of learning. They change their behavior in response to stimuli, right? So they can learn, but they definitely learn differently. Okay. So we actually have published this story, so I'm not going to go into a whole lot of detail. We know it's because of SAICAR toxicity that this happens, and we have found out that this is because of a lack of a molecule called tyramine that acts as a neuromodulator. So you might not have heard of tyramine. What tyramine is a biogenic amine, and you might think you've never heard of biogenic amines, right? But you actually have. So these are actually small nitrogenous molecules that are produced from amino acids, for example, tyrosine and tryptophan. Biogenic amines can be made from both of those amino acids. They act as neurotransmitters and as neuromodulators. They affect mood and behavior. And I said you've heard of them, all right, because this is dopamine and adrenaline and serotonin, and many people have heard of these molecules in the context of behavior and mood in particular. Okay, so we had determined that, yeah, all right, right here, we determined that when ADSL is gone, the SAICAR toxicity somehow interferes with the ability of tyramine to be made, okay? And that raised the question with us of like, well, is this specific to just tyramine, or is there something going on in metabolism here that's blocking tyramine as well as maybe possibly dopamine production? And could lack of dopamine underlie any of our phenotypes? And so that's a question that we pointed at next. All right, so this is back to that same movie I showed you a little while of the mutant. And what I want you to look at, you might remember that the wild type mutant, it thrashed back and forth. It went left, right, left, right, left, right. This animal, you can see it kind of stutter or thrash in the same direction twice. Here, 1, 2, right? And then it goes 1, 2 to the same direction. So it does this incomplete thrash. Wild type animals over here in the graph hardly ever do that. Most of them never do, right? A few might do it 5, 6 times a minute. However, ADSL animals can do this as many as 40 times a minute, right? So they have this weird behavior. We decided to investigate if dopamine might be involved in this by looking at mutants that don't have dopamine. So we took out from the worms, just like we take out ADSL activity, we removed the enzyme that's necessary to make dopamine, that's CAT2, and it caused the same phenotype, right? We took out the one to make tyramine, doesn't do anything. This causes the learning defect I told you about, but it doesn't result in this phenotype. And then in addition to removing dopamine from the control and showing that, hey, that causes the phenotype, we also can add dopamine back, right? Just by supplying it in the culture to the worms. And you see that you get a reduction in the amount of that incomplete thrashing that happens, right? So dopamine seems to rescue that phenotype. So we believe a deficiency in dopamine appears to be at least partially responsible for this uncoordinated behavior. So our model looked like this, where we think SAICAR is toxic and that was somehow inhibiting tyramine biosynthesis. And now we're kind of moving that upstream, and we think that, SAICAR is toxic and not just inhibiting biosynthesis of tyramine, but also of dopamine too, which indicates it probably acts someplace at a higher level to interfere with all of this, this tyrosine metabolism. And so that's the hypothesis we're working on right now. Okay. So basically we've linked lack of ADSL to some deficiency in biogenic amine signaling. This is another phenotype that we more recently how I've published. So this is about to come out. It's online already, but about to be published in Molecular and Genetics Metabolism. So we have found that when you overstimulate the muscles of ADSL1, they respond very poorly. So what we do is we flood them with levamisole. This is a neuromuscular junction, and this is the muscle side. And when you put levamisole on the animals, it activates the acetylcholine receptors in the muscle, causing contraction, and these animals paralyzed. It's a paralytic contraction, or yeah. Um, Contractile paralysis, that's what I meant. So these in red is the normal animals, and as they spend more and more time in levamisole, you see that they paralyze. ADSL animals also paralyze, but it takes them a lot longer, right? Even up to 10, 15 hours, you still have a small percentage that aren't paralyzed, although they do paralyze by 24 hours. So this indicates that their musculature can't respond normally. Interestingly, though, if we force the neurons to over, if this on the left, we are forcing the muscle to respond. On the right, we're using a different chemical, aldocarb, that's basically forcing the neurons to overstimulate the muscle. All right, so it's causing the same contractile paralysis, but it relies on the neurons to do it. Here we find again, and two animals, they paralyze quite quickly in aldocarb, whereas the ADSL, some of them never paralyze even after 24 hours. So they are very robustly resistant. What we have here in green is just a controlled different mutant that others have shown is resistant to aldocarb, and our animals are actually maybe even more resistant to aldocarb than what's been previously published. And so it's quite clear that the animals have both a postsynaptic problem, their muscles can't respond well to the signal, and they have a presynaptic problem in that their neurons aren't producing, it seems, enough of the signal to stimulate the muscle either. So we've identified problems at the neuromuscular junction. Okay. Okay. So basically that's the end of talking about three different phenotypes: an uncoordinated phenotype, a learning phenotype, and the neuromuscular junction phenotype. So now I said what we do is we take away ADSL from the worms, we see what's wrong with them, and then we're trying to see what can we do to try to overcome that. So that project coincides with this project that was started by your ADSL community, right? So I'm not sure how many years ago it's been now, it's been a few. There was a contract made with Perlara that investigates known or drugs that are already approved for use in humans for possible use in other rare diseases. And so ADSL did an experiment where they put drug libraries on yeast that were deficient for ADSL and asked which of these drugs seem to help those yeast grow better. They release that list. We take that list, right, and try to see, do they have any beneficial effects on any of the phenotypes we see in C. elegans? So basically, they did two different screens of two different drug libraries. 7 hits came out of the first, 20 out of the second, and their top hit, the thing that worked the best in the first hit was disulfiram, and the thing that worked the best in the second hit, in the second screen, sorry, was ara-uracil. And so we've been using these two molecules as well as some of the other ones that were identified to look at what they do in worms. So again, we have normal worms, we've taken away ADSL, we know they have abnormal behaviors and development. We add the drug and then we search through those behaviors and see if the drugs can have any positive effects. All right, so here's an example. This is it's a phenotype I didn't tell you about, but it goes right along with learning because this phenotype happens when you don't have enough tyramine. We know that, okay? So you add tyramine and it fixes this. So you can see the effect of the phenotype here. It's how they move their head when they're foraging. Normal animals all move their head. ADSL animals here in blue don't, okay? However, when we supplement the culture with ara-uracil, you can recover a lot of their activity, right? Actually, I said that backwards. These animals don't move their head. These animals do, but otherwise, this is normal. This is not. You can return them to normal by adding ara-uracil, by adding disulfiram, by actually adding fumarate. I didn't talk about this, but one of the byproducts of ADSL metabolism or the reaction is actually a release of fumarate. And so just by giving more of what ADSL should be producing seems to rescue this phenotype. Two other drugs that were in the screen, chlorambucil and malathion, don't do anything in this phenotype. So I'm not going to go through phenotype by phenotype. That is what we're trying to do. Next slide. There we go. Okay. But here's a kind of a grid of what we're doing. So basically, these molecules here in gray on the left-hand side are all drugs that came out of the Perlara screen. This isn't all of them, but they're the ones that we've worked with so far. Across the top here, you see a lot of different phenotypes, all right? The one that we've looked at the most is this tyramine-dependent phenotype. And we see that we have a lot of different uh, chemicals and products that actually can actually help reduce that phenotype. I wanted to point out down here, lamotrexol and methotrexate. These are two drugs we've seen two examples now of. Let's see, in Dr. SAICAR's talk, she talked about trying to reduce the production of that toxin using allopurinol, right? And Dr. Zikanova also talked about the potential toxin there, SAICAR. And so lamotrexol and methotrexate are both two drugs that block that pathway upstream so that SAICAR is never produced. And you can see that they both rescue this tyramine-dependent phenotype very well. We've also used these on the swim speed and crawling speed, and we actually do see some effect, but it's not as good as what we see with the tyramine-dependent effect. So it seems like these kinds of phenotypes might be due in part to toxicity, but probably there's other things going on as well. We haven't even begun to study the neuromuscular junction yet. I'm excited about those studies to see what drugs are doing there. And again, I showed you this data showing that dopamine seems to be playing a role in some of the coordination. Okay. So everything that's white on here is basically we don't know. And that is sort of what my lab is aimed at right now, is trying to figure out how to or to complete this grid so that we get some idea about which drugs are most effective where, which can give us some ideas about which drugs are doing what. Like, so for example, we think disulfiram is able to reduce the levels of SAICAR probably, which is how it's doing this. And so we're investigating those hypotheses. And then I just am going to end here. I get my slides to go forward there with some acknowledgments. So this is my lab. Not all these people, a lot of these are undergrads or students who've gotten their PhD already. So Maya in particular, right? And Sabrina are PhD students in my lab who are working on ADSL. And some of these other undergrads have had a part in this project as well. And then previous PhD student, Karina, and a previous undergraduate, Adam, played a big role in this project. And I want to thank the National Institute of Neurological Disorders and Stroke for funding for this project, and Penn State for funding some of the undergrads on this project. Also, Cure ADSS Foundation has provided some funding to my lab to study ADSS, and really appreciate just having the interactions with Rare Birds and Cure ADSLD people. That brings us into this community of people all aimed at solving the same problem.
Wonderful. Oh man, that was great, Wendy. I love seeing that final graph of all of the compounds and all of the phenotypes. That's amazing. It's such a dream of mine to come meet your worms and you. I love seeing them. It's so cool. Does anybody have questions for Dr. Hanna-Rose? One thing that I want to point out is you talked about Cure ADSSL1. Priyanka and Emma were going to come today. Don't know if they came in, but
Priyanka.
yeah. Oh, you did. Okay, good. So, oh, hi.
Hi, Nicole.
Hi. So I'm very fortunate. I was able to meet Priyanka at the very first Global Genes conference I went to. And I met her because I stood up in front of the whole place and said, I'm with ADSL deficiency. And she ran up to me afterwards and said, I'm with ADSSL1, which is something I had never heard of before. But since then, we've learned that we really we work with and share a lot of research and a lot of resources. And so it's been really wonderful to have Priyanka and Cure ADSSL1, as well as Eva with Cure ADSLD working together. I love these collaborations. They're so important to our mission. Um, so, so in terms of the dopamine, oh, uh, can you talk a little bit more about, or maybe if you know, like what does dopamine do in the body, and do you know that?
The answer is too big to talk about in an answer like this.
Yeah.
It does so much. So all of these biogenic amines um, can be neurotransmitters or neuromodulators. In other words, so a neurotransmitter like is for directly um, transmitting the neural signal from one neuron to another, right? Neuromodulators aren't necessarily involved in getting the signal there directly, but they actually change the level of electrical excitability of the neurons so that it's more easy or more difficult to send a signal to a neuron. And so they're everywhere in our brains, right? And involved in all kinds of processes. You've heard of dopamine, most likely in relation to Parkinson's disease, right? That's a disease where dopaminergic neurons are being destroyed. You've heard of serotonin, serotonin reuptake inhibitors are antidepressants, right? And so these things have biogenic amines and behavior and mood are very, very tightly linked, and their effects are complicated. Um, and I mean, this is kind of what got me into this field in even the beginning, because, for example, it's known in Lesch-Nyhan disease, which is another purine disease disorder associated with an enzyme in salvage synthesis, that dopaminergic regions of the brain are affected, but how that happens is still not known. And I thought worms might be a good system to try to figure that out. And so that's kind of how we landed here. But yeah, what does dopamine do? Everything.
Oh boy.
The nice thing about worms is are those behavior. So worms only have 300 cells in their nervous system. And we know all of them and we know where they are, right? And when they develop and everything. So we know which cells are actually important for these behaviors that we're talking about, right? So we don't know yet for the movement behavior, but we do know for the learning behavior exactly which cells are important. And so we have hypotheses about which cells are the ones that make tyramine and which cells are the ones that respond to tyramine. And in worms, you can do those experiments to actually prove it's this cell talking to this cell in this way. We aren't there yet, but you can do that.
Oh, Dr. Bordy?
Hi.
Hi.
Yeah, I have some questions, but let's say that I just try to just have one, summarize everything in one question. I was wondering if you if you check also glutamate level, or if you, let's say, assist that pathway in the nervous system of this worm.
Yes, we do have data. I'm trying to not confuse ADSS and ADSL data in my head here. TCA cycle metabolites are perturbed for sure. And the idea there that fumarate seems to have a positive effect. We need to investigate further and are using our LCMS to look at what's going on in the mitochondria, for example, with some of these drugs and some of these supplements. I know that you would be interested in that, and yeah, we should probably talk
As you
more
know.
now that I've had a chance to see your paper too. So yeah.
Okay, thank you so much.
Yeah, that was great.
One other thing there for Matteo, yeah, we have definitely seen mitochondrial defects in ADSS mutants in the muscle. So,
Oh, cool.
yeah.
Oh,
Awesome.
this is so interesting, these correlations. What I'll tell the group quickly before we go is that the previous year, Wendy had a grant to study ADSL, and that grant did not come through the following year, but the grant that did come through was for Priyanka's organization, Cure ADSS-L1. And I don't know how you're doing it, if it's just by the goodness of your heart, Wendy. But we're going to try and get some money coming to you next year for what we have coming down the pipeline. But you've been able to continue doing work and learning about ADSL1 also through ADSS-L1. And so again, this is why these collaborations are so meaningful and important, and we're so thankful. Thank you, Wendy.
Absolutely.